Uh, Very welcome, everybody. My name is Stefan Gunnarsson. I work at the Swedish Agriculture University, and I'm the coordinator of BroilerNet, and I will be the moderator today. And I will now share a brief presentation of what's going to happen today. Let's see here. So this is a webinar on avian influenza. Prevention is better than cure. And I would like to start to introduce our distinguished presentation persons here today. And I would like to start with Barbara Grabkowsky. She's a director of the Center for Sustainability Transformation in the area of intensive agriculture, and she works at Fechta in Germany. And she has developed an online check of farm level risk factors for avian influenza. And this was a good practice that was awarded the BroilerNet champion in the last cycle or the first cycle of BroilerNet. And then we also have Professor Jean-Luc Guerin, and he's a professor in poultry medicine at the National Veterinary College of Toulouse. And he's also director of the Host Pathogens Interaction Joint Research Unit. And he is a European specialist in poultry veterinary science. And Professor Natàlia Majó Masferrer, she's a director of the IRTA-CReSA, and she's also professor at the Department of Animal Health at the Autonomous University in Barcelona, and she's also a specialist in veterinary pathology and poultry veterinary science, I think. So, and this is the agenda of today. I will give a brief presentation of the BroilerNet project. Natàlia will present general concepts of highly pathogenic avian influenza in animals. And then Jean-Luc will present the current situation of influenza outbreaks. Where do we stand? We will have a short break at around 11 o'clock. And then Barbara Grabkowsky will present the online self-assessment tool. And then we will have a general discussion. And please put questions in the question and answer function of Zoom. And we will close this webinar not later than 12 o'clock sharp. So let me introduce the BroilerNet project. It's a project that is funded by the Horizon Europe, and we are 25 partners. It's 50-50 with academia and industry, and 13 countries within Europe is involved. And we are a project that will run for 4 years. So we have yet another year to go. And if you scan the QR code, you will get the LinkedIn account of BroilerNet, where you can find information and news and things about our good practices, etc. And you also have the link there. And we will put the link in the chat as well. So this is a picture of all the 25 partners of BroilerNet that are helping us and contributing to make the broiler production in Europe more sustainable regarding animal welfare, animal health, and environmental sustainability. So these are the focuses of BroilerNet, and we are focusing on what is happening to the farm. So it's important that we have the broiler farm and the broiler farmer in the main focus. And what we have been doing is to create bins that should not be considered to be dust bins, but it's broiler innovation networks in the 13 countries. And these are groups of farmers, advisors, and other stakeholders. And they have been guided by or are guided by facilitators that we have been training in a special method. So what the broiler innovation networks are doing is to find sector challenges, things that are important to the broiler farmers in the different countries. And then these challenges, we want to find good practices to mitigate these sector challenges. So an interaction with the broiler innovation networks, the BroilerNet coordination group, and then we have 3 thematic expert networks, of which one animal health management is coordinated by Natàlia. So what we have been doing, this is from the first cycle, is that from all these challenges that are reported from different countries, the thematic networks and the bins in interaction make a priority of the top challenges. And this has been done or will be done for 3 cycles. We collect good practices from different countries. For example, one country have considered that there is a big challenge with let's say, footbed dermatitis. Another country could contribute with ways of reducing footbed dermatitis, etc. So back to this picture, what we're doing then is to evaluate the good practices that come in. It's a lot of good practices that are sent in, and we evaluate them, and then we find the best of the good practices, 5 for each challenges, and it's 5 for each cycle as well. There is made a cost-benefit analysis of the one that are the top ones, and these will also be awarded as BroilerNet champions. And then we disseminate for use of the farmers and advisors in the different countries the good practices and the reports and material we have. So it should be accessible for European poultry farmers or broiler farmers. So in total, just to give you a view of how many good practices we get, is 45 good practices were collected in the animal health management work package. And these were evaluated, and for the challenges that were awarded a champion, and for fighting avian influenza, this self-analysis of risk factors, which Barbara will introduce to you after the break, is was the top one in this cycle. And then we have the Broiler Knowledge Hub, and you have the QR code, and you can find here all material that we have collected and developed. So you have fact sheets, videos, abstracts, etc. And the fact sheets are also available in some of the national languages, in case that the broiler farmers or advisors are not really happy with reading English. But in many cases, these good practices are available in the larger languages, which is not including Swedish, I would say. So here you see that they could be developed. They are all in English, but they are also in Italian and Finnish, etc., etc. So back to the agenda of today. It's a pleasure for me now to present Natàlia Majó Masferrer from IRTA that will give a presentation of general concepts of avian influenza in animals. So I will stop sharing and I will give the sharing to Natàlia. So
yeah,
Natàlia, please.
Good morning, and thank you very much, Stefan, for the introduction, and thank you everyone for joining us in this webinar. I will start by sharing my presentation. Let's see if it works well. Yeah, I guess you can see it now. Is that okay?
Yes. There is
Okay. Perfect.
a time lapse of, let's say, 2 seconds or something like that.
Okay.
But no problems. Please.
Perfect. Then, well, as Stefan already explained, I will cover in this first talk of the webinar the general concepts, the general principles of this disease in animals, and I will focus particularly in birds. And oops, sorry, I went. And I would like to start my talk with this map, okay? That is, I think it's the reason why we are here today in this webinar. This is the current situation that we have regarding avian flu. In fact, these are all the outbreaks that have been reported since October 2024 of zoonotic highly pathogenic avian influenza in different animal species. So this is the current wave, the current epidemiological season that we say. And as you can see, well, we have a major problem. We have a virus, which is mainly the strain H5N1, that is present in different continents, is in almost all the continents, I would say. Especially in Europe, we had, and Jean-Luc will talk more about this later on, but we have a lot of cases in wild birds and in poultry. Also in US, the situation is quite complex and worrying, I would say, with virus in wild birds, poultry, and also in cows, as you probably know. And then I would also highlight the presence of the virus in the Antarctica. Okay, never before a highly pathogenic avian influenza virus have been detected in this continent, in the Antarctica, and last year it was detected causing mortalities in wild birds and also in seals in this continent. So the conclusion is that we are now facing a major problem with avian flu, not only in terms of, or for the poultry industry and the livestock industry, but also in terms of biodiversity and also public health, because as we mentioned, these are zoonotic viruses and they can spill over and cause disease in humans. So, but let's start from the beginning, and this is our main player here. This is the avian influenza virus, which belongs to the family Orthomyxoviridae. And this is a huge group of viruses that are divided in 4 different genera. And we will focus in the influenza virus type A, that are the only ones that are infecting birds. And I would like to highlight 2 characteristics of this virus. The first one is that this virus is presenting with a segmented RNA genome. And as we will see, there are 8 different segments and they encode for different viral proteins. But the most important, and that you have to keep in mind, is that this virus can reassort and can combine. The segments can combine and create very easily, as we will see later, new viruses. And then we have these 2 viral proteins which are also important. We have the green one, which is the hemagglutinin, and the blue one, which is the neuraminidase. The hemagglutinin is the protein which is responsible for the viral entry into the cell. And the neuraminidase is the protein that facilitates the release of the viral infectious particles from the infected cell. So far, there are 19 different types of hemagglutinin and 11 different types of neuraminidase that have been isolated from different animal species. And there are many different combinations of these proteins conforming the different viruses. But how are these different subtypes in the nature, distributed in the nature? The most important is the role of the wild birds. The wild birds, and specifically the waterfowl, are the main reservoir, the main natural reservoir. And actually, all the possible combinations of hemagglutinin and neuraminidase have been found, have been isolated in different waterfowl and different wild bird species. And from these wild birds, the virus can spill over to other domestic poultry, for example, or even a different type of wildlife animals, for example, wild birds, seabirds, different carnivores, marine mammals are also affected with specific subtypes of viruses. And now we also know that they can infect ruminants in the domestic counterpart, horses, pigs, and in humans, there is a limited number of viruses that may infect humans, but as you know, that zoonotic events may also occur. And I would like to show this paper, these results that are quite old, but I think they exemplify very well. This is a study with a large number of samples. In this study, they sample thousands of wild birds, and they demonstrate that the Anseriformes, the order Anseriformes, and more specifically the ducks, are the ones that show the highest prevalence of this viral infection. But how? I mean, we know that the influenza virus is really diverse. We know that we have to change the vaccines for humans each year. But how is this viral diversity achieved? I mean, how these new viruses emerge every now and then? We have two main mechanisms of diversity. One is the antigenic drift, and these are single mutations that occur in one specific virus. And this is because the proofreading mechanism of the polymerase is not present in this type of viruses. So they can accumulate mutations. And if the mutations are located in a specific antigenic sites, for example, of the hemagglutinin, these new viruses may not be recognized by the immune system, and they may cause a different clinical picture. Okay. And then we have another mechanism, which is important, which is the antigenic shift. And the antigenic shift means reassortment of the gene segments between two different influenza viruses. And this is causing sudden changes, and there are important changes. Okay. How this may happen? We know now that there are some animals, like, for example, the pig, that has receptors for different types of influenza viruses. And so they can get infected, for example, with an influenza virus coming from birds and another one coming from humans in the same cell. And there, the different gene segments can reassort and create a completely new different virus. This has happened before, and is the cause of the several pandemics that have been described in the last century and in this century as well. So another characteristic of the influenza virus infection in birds compared to mammals is that there are two different clinical pictures. We have the low pathogenicity clinical picture, and this is the birds are not showing any clinical sign or very mild clinical signs, respiratory or even mild diarrhea, and there is no mortality. And so the infection is restricted to the respiratory and digestive tract. And we have also another clinical picture, which is the high pathogenicity clinical picture. And here we have an acute systemic disease, and it's causing 100% mortality in a very short period of time. And the animals, in this case, show a systemic infection. And which is quite interesting, is that only the subtypes H5 and H7 may cause this clinical picture. So what do we have is this situation that we have the low pathogenic viruses in the natural reservoir, in the waterfall, and then by exposure, these low path viruses adapt to the domestic birds. These viruses are circulating in the domestic birds, and they can have mutations, and if these mutations are in the hemagglutinin site, then they can become highly pathogenic and cause 100% mortality, high mortality in the domestic birds. And what is also important is that these highly pathogenic usually do not readapt to the natural host. So, and how can we explain these low pathogenic and high pathogenic characteristics of the influenza virus? What we saw is that this is because there are differences in the cleavage site of the hemagglutinin. The cleavage site of the hemagglutinin is different in the low path and the high path viruses. So we have the precursor protein, and in this precursor protein, the protein has to be cleaved. And in the low pathogenic avian influenza viruses, we have a monobasic amino acid in this cleavage site. So this cleavage site will be processed by trimseed like proteases, and these enzymes are present only in the cells of the respiratory or digestive infection. So when one animal is infected by these low path viruses, they only replicate the virus in these tracts, in the respiratory and digestive tracts, and they have a mild clinical outcome. Whereas the highly pathogenic viruses have a multibasic cleavage site, and this is cleaved by furin-like proteases. And these enzymes are present in all or many tissues in the birds. So they get a systemic infection, very lethal and with high mortality in this bird species. This is an old picture, but I think it's very representative of what happened when highly path avian flu is hitting a farm. This is a turkey farm from an Italian outbreak in 1990s. And you can see here that very rapidly, almost all the birds are dying because of this infection. And regarding the macroscopic lesions caused by the high path viruses, what we have are the lesions which are a result of the replication of the virus in the tissues. We said that the infection is systemic, so the virus is causing viremia quite rapidly after several hours after infection. And then they go to different tissues. And one of the main tissue affected is the pancreas. And the virus is causing there replication and necrosis of the pancreatic tissue. And here you can see the necrotizing and hemorrhagic pancreatitis, which is typical, very typical of high path avian flu. But also the virus is replicating very characteristically in the vessels of the birds. So, and they have this these characteristic hemorrhages in the skin and subcutaneous tissue. This is also very characteristic of the high path avian flu. Sorry, I go too fast. And wait. Okay. Here are more pictures. These are microscopic pictures, and that showed what I just explained, that the virus is replicating in different tissues, in many different tissues. For example, here we have the pancreas, we have here the necrotic areas, and the virus is replicating, and we see it with this immunohistochemical staining. Organs as different as the pancreas, the thymus, the liver, spleen, muscle, and also the central nervous system. And actually, the central nervous system and the replication of the virus there is very important. The high path avian viruses are really neurotropic. So they reach the central nervous system very early after infection. And then they distribute widely in all the nervous tissue. Here we can see, this is a diagram. This is a paper that we published several years ago. And you see that at 2 days post-infection, these gray areas are the areas where the virus is replicating. And by 4 days post-infection, I would say that more than 50% of the nervous parenchyma is infected. And this is the major cause leading to death in this animal. So this is very characteristic for the high path viruses, the neurotropism that they have. Regarding the transmission routes of avian flu, we have the low path viruses that are transmitted mainly via fecal-oral route. the animals that are infected, the natural reservoirs, the waterfall, they shed large amounts of virus in the feces and then contaminated the water environment. And then other animals, other birds may ingest this water, this contaminated water, and get infected. Whereas the high path viruses, the routes of transmission are more complex because, as I said, they are shed by not only by the respiratory route and the fecal route, but also there is a lot of viruses in the viscera. And for example, scavenger birds that can feed with these infected carcasses can get infected by oral route. Some data on this persistence and transmission of the viruses. For example, for low path, it is important to know that the persistence of the viruses in the water can be really long. And these are experiments that we carry out with different viruses, low path and high path viruses, in different conditions, different temperatures, and different salinity content of the water. But you can see that the virus may remain infectious until several days, or more than 90 days in some cases, in the water. So this determines that the ecology and the persistence and the infection of the virus in the natural reservoirs. And for the high path, as I said, one of the ways of infection is the ingestion, is the oral route of infected animals. And here we perform a study where we check the infectivity of different tissues in the animal once the animal is dead. And for example, in a skin, the skin is infectious even 6 days post-mortem. So the virus is really persisting in the infected carcasses, and so other animals, other birds can get infected from eating these carcasses. And also, quite recently, and together with this is an experiment that we did together with a group of Jean-Luc, and we determined that the feathers can be also or can play a role in the distribution and dissemination of the virus, because the virus is replicating quite well in the epithelium of the feathers. So this may explain also the transmission between the birds in a farm and among the farms. So, well, till here, I've been talking about the classical aspects or the classical characteristics of the avian flu. But it's true that with the current strain that we have, with the H5 Guangdong strain, that is the one that is circulating now over the world, there is a new scenario because some of these characteristics have changed. And I will focus now my talk on these differences that we are facing. So this particular strain, the H5 Guangdong lineage, emerged in the 90s, in 1996 in Hong Kong, but actually it was circulating silently until 2004, where high mortality in wild birds were reported in China. And then via the migratory route, via the wild birds infected, they enter into several countries in Europe. So the first epidemic wave or epidemic outbreak, important outbreak of H5N1 in Europe was in 2005, 2006. And since then, there have been several waves of this viral infection in Europe. Also, the virus has been reassorting with low-path avian influenza viruses that are present in the wild birds residents in Europe. And I would say that since 2020, the virus is circulating in different levels, but quite continuously in many countries in our continent. So which is the main difference of this virus is that the high path viruses is not restricted now to the domestic chicken, to the domestic birds, but it's already present in the wild reservoirs. So we have wild birds which are infected with high path viruses, and this is an unprecedented situation. Has never happened before that highly path viruses can be found in these wild birds. And this is quite nicely shown in this diagram that is from the World Organization for Animal Health, that it explains very well which was the historical scenario. So in the past, we had low path in wild birds, and they spill over to the farms, where they mutate in the farms in domestic birds, and then they spread to other farms. But now, with this newest scenario, with this new H5 goose guangdong lineage, we have the highly path in wild birds already. So we have low path and highly path in wild birds. And from here, it's very easy, because it doesn't have the virus. There is no need of mutations because it's already high pathogenic. They can spill over to the farms, infect the farms, and spread to other farms. But it also may spill over to other animal species, and particularly mammals. And this is a map showing all the cases that have been reported of mammal infection around the world by this strain. And you see that the diversity of mammal species that are affected is really high. Actually, till now, more than 50 different species of mammals have been infected with this strain. And well, you probably know several outbreaks that occur, for example, in marine mammals linked to this H5N1, the one in Chile, for example, that thousands of sea lions were dead because of the infection with this virus. Also, farm animals, fur farm animals in Spain and also in Finland got infected. And here, the transmission from animal to animal cannot be ruled out. So that's quite concerning. And also another characteristics which has never been seen before is that in all these mammal species, there is a new neurotropism, because before the virus in mammals didn't infect the central nervous system, but now in all these cases, or in many of these cases, we see that the virus infects the central nervous system. Well, and finally, I'll talk a bit about the outbreak in cows in US, which is probably the newest new host that this high path viruses has infected. This was in March last year that the first cases in dairy cows were detected, with no clinical signs or very unspecific clinical signs. There is no mortality, but in the cows, there is a high viral load that is present in the milk. So nowadays, we have more than 1,000 farms in the US which are infected in 17 different states. And the most alarming is that the virus is also causing disease, or although it's mild, but it's causing disease in humans. So in several farm workers, the virus has been found causing mild conjunctivitis. So we will have to keep an eye, because the outbreak is still ongoing, and we'll see how it evolves. So, well, summarizing, the avian flu is a major threat today. I would say not only for the poultry and the livestock industry, but also in terms of biodiversity, because the host range of wild birds and wild carnivores and mammalians is that it can be infected are increasing every day, and also for public health. And I just mentioned the cases that we that are seen in the United States. These circulating strains are becoming endemic in several countries, and particularly in Europe. We have cases now almost all the year. There have been major changes in the epidemiology. As I mentioned, there is a broadening of the host range. Now we have wild birds and mammal infected, and this neurovirulence in the new hosts is also a characteristic of this virus. So I would say that the continuous surveillance and early detection of the virus is essential to control the disease. And probably today we will learn more about these control measures that can be taken to control the viruses and prevent the further transmission and adaptation to this new species, which is also, I think, very important. And well, I would conclude my presentation here. Thank you for your attention. And if you have any question or comment, I will be more than happy to answer.
Thank you very much, Natàlia. It's not possible to give an applause. And I see that in the webinar function of Zoom, it's not possible to send flowers, etc. So, but thank you very much. And please, if you have questions for Natàlia, put them in question and answering function. And I hope that we will get some of them. I have a first question for you, and that is, the trend looks quite scary for the future. Yeah. I mean, what is the prediction? Will there be a possibility to take down the curve because you see an upward turning curve? What do you think?
Well, we don't know actually, and here it will be nice to hear different opinions, but I think that that the situation is quite alarming. I agree with that, and I think we all agree with that. And we don't know how this virus will behave in the future, because it has changed completely the paradigm of avian flu. All what we knew from the past is not applied not right now. So, for example, 1 year ago, nobody thought that the virus could infect ruminants, and now we have a huge outbreak of avian flu in cows in US. So it's very hard to predict, but the situation is not, it's not positive at all. It's not positive at all. And also the capacity that this virus have to adapt to new hosts, and especially humans, is quite worrying. Yeah.
Yeah, there will be an oral question here. I will from Pritam Kuhad. Please. Can you please tell your question?
Ma'am, where do you see the uprising trend of antimicrobial resistance in the upcoming years?
Wow. This is not related to avian flu. Well, of course, antimicrobial resistance is also another threat that we have to face from the veterinary perspective. What? Sorry? Oh.
Sorry, ma'am.
And I think that for the poultry sector, I would say that I guess at least in Europe, there is we are working hard to reduce the use of antibiotics. That's for sure. So we are fighting and contributing to this reduction of antimicrobial resistance.
Thank you very much.
Thank you very much, ma'am.
Lina, do you find another question?
No,
No?
can't see any.
Thank you very much, Natàlia. There are
Okay.
more chances to ask questions, but now we need to go to the presentation by Jean-Luc Guerin. So, Jean-Luc, please. We can see your presentation. And it's soon in presentation mode.
Okay.
Yep.
Is it
You're welcome. Yeah.
Okay.
Maybe it's a time lapse there, but if you press, yeah, we get it in your presentation mode with the speaker mode.
Okay. Okay.
Yeah.
So, sorry, I will double check. And I will try again. Sorry for that. Anyway, if it's the same, I will keep like this. It's not a big deal.
Yeah. Yeah. Please.
Okay.
You're welcome.
Okay. So many thanks for the invitation. And actually, I will try to share some information and data and in complement to the presentation of Natàlia. Regarding the current situation at the global scale and at the European scale, I will basically, I'm French, so I'm sorry for that, but I will propose also a small focus on the French history and the evolution to the vaccination strategy, and I will finish on the options, including vaccination at the European scale. So, as was mentioned by Natàlia, we are just facing a very, very major situation, a major challenge with a family of viruses, a very specific family within the avian influenza viruses, and one clade, the clade 2244B, is now the major challenge we're facing, at least in Europe and in North America. It's really, really important to realize that this is the outcome of a long-term history of almost 30 years of genetic evolution. That's why we need, as soon as possible, to stop as much as we can this type of genetic evolution. The global situation is this one right now, with the global majority of viruses refer to the H5N1 subtype, sorry, oops. And with circulation in the North Hemisphere, in Europe, and now in America, but also in Africa, and different clades are still circulating in Southeast Asia. I will do not develop this situation, but we should not forget that we are there. I would say in the nest, in the original scene of emergence of these viruses, and we are still cases of other clades, still cases in humans, still deaths of humans due to I would say the original or more ancient clades of viruses. And if we consider the curve of cases, we see that we observe globally the same situation. This is the curve of cases observed in the northern hemisphere. I mean, in Europe and in US mostly. And we observe this evolution. And what you see is that obviously the situation in 22-23 was a disaster. The situation last season was much better, and this season is almost intermediate, I would say. And we observe the same evolution at the global scale with a slight decrease, but we still observe cases, a few cases, even in the summer period. And that's very important. Since 2022, there's no stop during the summer season. And we'll see that this is linked also to the endemic and zoonotic circulation in the white bird sector. And if I go to a white bird situation, this is the 5-year history of outbreaks detected in white birds. And you see that we observe some waves. Obviously, here again, the circulation was absolutely huge since 21 to 23. And then we observe some decrease, but almost non-stop circulation. That's very important. The fact that there's non-stop circulation, there's no season where we do not observe any circulation. And this circulation is almost widespread at the European scale, with a global majority of H5N1 genotypes observed at the European scale, with a huge diversity of species of birds. That's also a very important point. We of course observe cases in waterfowl, and as Natàlia explained, it's completely consistent with the biology and the long-term history of avian influenza. But for the last 2 years, the contamination of colony breeding seabirds has been huge, with an adaptation of specific genotypes in these birds and a wide circulation in these birds. And we also observe circulation in other birds, for example, raptors, and obviously these contaminations are obviously linked to predation of infected animals. So this diversity of species is very important, because that means that you could observe an endemization, long-term circulation, not dependent of migration. That's important. And as an indication of this circulation, I would like to illustrate this with some investigation performed on cats. And the main question is not the health of cats, to be honest. It's only that cats are sentinels of exposure to some kind of birds, and mostly small passerine birds. That means that if I go back to the previous picture, we mostly detect during the surveillance, I would say, big birds, ducks, swans, etc. And we are pretty sure that we do not detect the circulation, the low circulation in small birds that are much, much harder to identify. And the importance of this type of investigations in cats is that they suggest that we are still facing a circulation of virus, an endemic low circulation. A paper has been published last year by colleagues from Utrecht, and we released a paper just a couple of weeks ago on a survey performed for more than 1 year in France. And this information suggests that cats are exposed globally to this circulation. And the result is a non-stop introduction of virus in farms. And the mechanism is this one. You have circulation in white birds and sometimes circulation in poultry farms. And depending on the type of farms, depending on the density of farms, you could have more or less farm-to-farm diffusion. That's the global mechanism. And for the last months, I mean, you are here the very last period in the official reports. You see that we still have circulation in captive birds, I mean, small-scale conservatory farms and commercial farms in red dots. And you see that we are still always cases. And the distribution of cases is interesting to consider. We see that we have a lot of cases, reports in UK, in Italy, in Poland, in Hungary. And in the last weeks, we have very strong circulation and identification of cases in Hungary, for example, with a strong focus on geese and ducks farms, which is quite, I would say, consistent with a very high sensitivity of these birds. But we see that the impact is very important in Poland. The diversity of poultry species is very strong. We always identify cases in ducks, in geese farms, of course, but also in turkeys and in broilers and in layers. And the economic impact of these cases in layers is obviously very important. And we also observe a global impact of cases in breeders. And I would like to emphasize this. The impact on breeder production is very huge, since obviously you will have a second effect on the production of broilers themselves. And you see here a tentative of distribution, of description of the different types of birds. I mean tentative because in black you have here the unknown species. And if we go further to try to identify the situation, we know that there's a mix of different species in different countries, between waterfowl, between turkeys, and between gallinaceous birds, I mean broilers or layers. Just to focus on the specific history of France, because I mean, I think it could be illustrative of the different risks that we have to control. What we observe for now almost 10 years is that we could have different mechanisms. We could have emergence of H5 viruses from low-path viruses. That was the situation in the very first outbreaks in 15, 16. And then we observe, like other countries, the introduction of a clade 2344b. And with a strong focus on duck production in the first years in the southwest, and it was linked to very specific ways of production, techniques of production with outdoor breeding. But in 22, we also observed waves of outbreaks in Pays de la Loire, in the western part, with most of farms raised strictly indoor. And then a diffusion of these viruses. And what we showed is that the density of ducks in the French situation is a very strong risk factor, a very strong risk factor. And these modeling investigations leads to the creation of a specific zoning in yellowish here of zones with high density of farms. And this is very specific to the French situation. And 1 year after the creation of these zones, we observe a merge between this prediction And the case is observed in 22 in Pays de la Loire and in the southwest. And this merge means really that the control of the duck sector was critically important. And that's why the vaccination strategy was targeted on this sector. So if I go back to the mechanism and to the presentation of Natàlia, we know now that we have to consider different risks, the risk of introduction from the white birds, and once the first introduction has occurred, the most important point is to control the farm-to-farm circulation by airboat, by all ways of connection in the poultry industry. But now we do think that the role of the commensal birds is, I would not say marginal, but it's not central in the farm-to-farm distribution. It's really in the end of the poultry industry. And it's very difficult to control this because you all know that the ways of introduction and contamination from farm to farm are almost impossible to control, It's very, very difficult, at least in high density area. And I cannot develop here, but depending on the types of production, if you have broilers plus ducks, broilers plus turkeys, you will increase the risk of dissemination in the given region. So the response to outbreaks are very important, but they show limitations, obviously. The classical strategy is based on surveillance, biosecurity, and depopulation of outbreaks as fast as possible. And it's still critically important. So I think that we will discuss this in the next presentation. It is still critically important to have a clear map of the production, a clear map of the fact, and to be able to detect as fast as possible and depopulate as fast as possible any outbreak. But we know that at least in high density zones, it will remain not totally efficient. And we observe issues based mostly on the capacity of culling and disposal and the extremely high transmissibility of this virus. So almost 3 years ago, the discussion started really, I would say, seriously regarding the evolution of a global strategy. The postulate is that the basis of any control of this virus is and will remain based on biosecurity and surveillance. And the discussions was, okay, could we include vaccination to the toolbox or not? And I will not develop here, but you all know that it's not that easy, and the balance between pros and cons has been very, very difficult, and is still a matter of debate. But anyway, at least in the French situation, we consider that the situation was not sustainable anymore. And globally, the postulate evolved quite strongly with evolution of a policy of at the global scale of animal health organization, at the European scale. And we try to address different issues, technical issues. For example, the choice of good vaccines. I will not develop here, but we identify some vaccines that are efficient and that allow discrimination between infected and vaccinated animals. And some trials have started in 22 in ducks in France, in turkeys in Italy, in layers in the Netherlands. And so far, to date, France is the only country that actually started a large-scale vaccination program 2 years ago. And this strategy was based on vaccination of ducks at the production level, ducks only, optional for breeders, and prohibited for all other types of poultry. And this strategy has been based along with a very strict surveillance program. And I just put here the regulation. The EU regulation right now is based on this type of surveillance. It's very strict, it's very expensive, by the way, but it's critically important if we want a success to associate the vaccination plan to a surveillance. And very quickly, we a preliminary investigation performed a few months ago suggests that the vaccination in the French situation changed dramatically the number of outbreaks. In the lower part, in the left here, you see that you have here the prediction of cases. And here you have the actual description of cases during the last period, I mean, the period between 23 and 24. And we see that the prediction is more or less consistent with the actual description of cases in different countries, but not in France. And we should have observed between, let's say, 300 to 700 cases, and we actually observed 11 cases in the period. And in the last months, we can say that it's pretty the same situation. We can observe a dramatic decrease in the number of outbreaks observed in France. I insist on the fact that it's really country dependent. It's dependent. It's based on the structure of the production. But at least in our experience, the vaccination has dramatically improved the situation. And who's next? We know now that the situation is evolving in the Netherlands, not in ducks. Obviously, there's almost no duck production there, but in layers. And we know that the situation and the cost-benefit analysis in layers is very difficult due to the economic impact of outbreaks in this production. So last month, the field implementation started in 1 layer farms, and we'll see the evolution in this country. So obviously, avian influenza will remain for years and years a threat to the poultry industry at the global and the EU scale, obviously. In my opinion, there's no reason to keep a taboo on vaccination, provided we implement seriously with a very strong association with classical biosecurity and surveillance. The vaccination programs should be shaped to all situations and mostly on different poultry production. The surveillance is a key. There's a room for investigations and for modeling studies to shape the strategy and with or without vaccination. And as Natàlia mentioned, this is absolutely not the end of the story. The virus is still evolving, is still adapting to new hosts in birds, in mammals, and it will remain for some years a very daily challenge. And with this, I would like to thank again the organizers for the invitation and you all for your audience and my group and team for their support. Thank you.
Thank you very much, Jean-Luc. It's not possible to give an applause, but thank you very much for an interesting presentation. There is now time for a couple of questions to Jean-Luc. So please put them in the question and answering. Maybe I didn't really follow thoroughly, but what is the vaccination efficiency of these vaccines in general? Is it 99.99% or?
Yeah, it depends. It depends mostly on the target species. I mean, we know that it's much easier to provide a very efficient protection in chickens, meaning broilers and layers, than in ducks or turkeys. It's due probably to specificities of a immune system. And we also know that, for example, in chicken, you have very efficient vaccines that can be implemented at the hatchery. It's much easier. But the assessment of efficiency may be very tricky. And we have to be very careful because you may have, let's say, 70–80% of efficacy at the bird level, but a very strong and 100% impact at the farm level. We have to keep in mind that it is a population vaccination. And we have to consider this as a population vaccination. So it's very important to consider the experimental trials, but also the field impact. And that's all the input of modeling studies. We don't need a 100% protection to provide, or at the bird level, to obtain a 100% protection at the farm level. And we cannot extrapolate information from chicken to ducks to turkeys. And we cannot use exactly the same vaccines in all the species. That's the tricky point regarding vaccination.
Thank you. Lina, do we have any questions? Maybe we can have one because we need to have a short break soon. And I mean, then we save the question for the next session.
Yes, there's a couple of questions. One is, what are the consequences in international trade if the country is vaccinating?
Yeah, I expected this question. That's why I deleted this slide. That's still the key issue. And that's why it's very important to be convincing on the fact that we keep the objective to control the infection. And so the impact for France has been, I would say, limited, but it has been limited because of a very strong work performed by the French administration. Months and months before the implementation of the vaccination, and since then, and it took some months of struggle to convince some key countries to, let's say, to release any ban on the production. But that's why it's critically important to associate vaccination and specific surveillance plan. Because if you don't proceed like this, first, you will not be able to control on the field. And that's the very important point. And second, you will not be able to convince commercial partners that you are actually controlling. So, there's a lot of work that is still to do to improve the cost benefits, let's say, balance of surveillance plans. But that is very important. So the short answer is that the impact has been quite limited. It's still a struggle, And we still need to work on vaccination plan and surveillance plans.
Thank you. Lina, I think we save the other questions for the general panel discussion because it looks like there are both for Natàlia and Jean-Luc and maybe for Barbara as well. So we will have a 5 minutes break now, and please be back at 11:15. And then Barbara will give her presentation. And then in the end, we will have a panel discussion with the questions that you have brought in. So see you in 5 minutes. Thank you very much.
Yeah.
Hi, Stefan. Can you already see the slides?
Yeah.
Okay, it's in the right mode, right?
Yeah, absolutely.
Okay.
Absolutely. Yep.
Very good.
Yeah. And it's 11:15 now. So Baba, it's a pleasure to have you here, and you were, and the Good Practice was awarded the BroilerNet champion of the first cycle. So it would be very interesting to that you give a presentation. And I also put in the link to the BroilerNet Knowledge Hub for those of you who want to look at the good practices, and there you will also find this good practice. So, Baba, you're welcome. Please.
Thank you so much, Stefan, for the kind introduction. So good morning, everyone. It's great to be here and to present our online self-assessment tool for farm level risk factors. Yeah, what I would like to talk about this morning, ladies and gentlemen, I would like to give you some ideas about the objectives of our farm risk tool. They will have a short glimpse on the methodology. I will present, of course, the tool itself, and I will finalize with some conclusions. Yeah, let's start. What we heard from Jean-Luc and Natàlia this morning is that we have a new dimension. So avian influenza is a panzootic, and so we need to take into account that it's a year-round risk of introduction and further spread. So it's no longer seasonal, and that is what especially farmers need to take into account. And that we have a new situation that we have low pass and high pass viruses that occur in wild birds. But we also have to take into account the tenacity. Jean-Luc was also referring to it. So the stability of the virus in feces, in blood, in any contaminated materials needs to be taken into account. So sound cleaning and disinfection is really, really essential when working on farm level. So we need to have establish a continuous awareness and prevention, and in other really basic, but of course problematic topic is that we cannot see the virus with bare eye. So, and there are many routes of infection. So we had already a picture from Jean-Luc, but I just brought a figure what kind of different introduction pathways we have only from people that can bring the virus to your farm. So, and it is up to your responsibility how big are these gaps to enter the virus into your premises or in the poultry house, or if you can manage to make the gaps and the weak points as close as possible. So the farmer actually is in the responsibility, and we need to have a professional and consistent prevention management across all the livestock systems. So the monitoring, what was already referred to, but we need to have a 24/7 biosecurity by the entire team working on the farm, but also along the whole supply chain. So we need a farm-specific, systemically designed concept, and this also actually refers to our European animal health law. You may have seen there was an update. It is already longer in place, but here in Germany, it's a hot topic at the moment because it comes into effect and force, especially when talking with the animal disease funds. So they all the livestock farms need now a plan for protection against biological hazards, and farmers increasingly bear the responsibilities because they are interpreted as entrepreneurs who are in the responsibility to take all measures to prevent any incursion at their farms. So there are, of course, a lot of checklists, recommendations, and papers how to prevent viruses to come to your farm. But the thing is that, of course, there are numerous checklists, but we often face the situation when going to farms that is little consideration of the farm-specific risk profile. So I work as an auditor some years, and when you go with all the different risk factors through with the farmer and they recognize it, they say, yes, I have, yes, I have, yes, I have this. But really a reflection, what is at the individual situation does often not take place. So that is why we wanted to have an individual farm level risk assessment for farmers, but also advisors and veterinarians. And therefore we came up already so 2018 with an online tool in collaboration with research and practice here in Germany. So there are a lot of risk factors that we took into account for a virus introduction. And we wanted to have like a structured function-specific analysis with not more than 100 questions, and that it should not take longer than 1 hour. And the results shall be available directly upon completion. So, and it is, of course, free of charge. It is anonymous, and it shall reflect the individual farm risk profile and offer tailored optimization suggestions. So, as I mentioned before, so we started with AI because I worked, my PhD wasn't about avian influenza, and I worked a lot in the poultry sector. And therefore, we started in 2018 with our farm risk tool for AI. We adapted this to the animal health law. Then we also have it for African swine fever and free-range farms. And in the beginning of this month, we launched a farm risk for ruminants. So, and especially in the light and the foot of mouth disease outbreaks we currently have in Eastern Europe. There's a lot of interest currently to raise the level of biosecurity also on cattle farms. And we're also working on a farm risk tool for small ruminants and also for agriculture, just for letting you know. So talking about our steering group, so we really intended to have a multi-actor group. We had a steering group with the product board here in Lower Saxony, but also the product board, the poultry product board from Germany. Based in Berlin. Then we had the Friedrich Loeffler Institute, it's a federal institute, and also a member of the Lower Saxony Animal Disease Fund, because they are, of course, and I can refer to it later on, actually talk with you about the compensation when you have not complied 100% to biosecurity, then probably your compensation will be less than 100%. Yeah, and we also work with QS, quality and security, which is our quality assurance scheme here in Germany. Yeah, so let's have a short look at our methodology we used here. So there, as I mentioned, there are many risk papers and recommendations and checklists, and we started to gather them all. We hope we gathered the most important ones. We use the EFSA opinion and many journal papers and so on, and gathered a list of all the risk factors for an incursion of avian influenza. And then we established a multi-actor panel with more than 60 people throughout Germany, from veterinary practice, of course, a lot of farmers, a lot of state veterinarians, and different people who checked in the first stage this list, whether the list is complete. And then we did a Delphi study. Here you all the participants of the expert panel weighted the single risk factors according to the epidemiological relevance for the introduction of a virus in a farm. And in the second stage, they did this again in the light of the descriptive analysis or statistics from the round one. And with this procedure, this is very well documented and very well published. We gathered the weights for every risk factor, then we adapted it in our online system and programmed the online system, and then we made a first draft actually. So what kind of topics and risk factors did we consider? So we have actually 7 different chapters. So we have, of course, general information of the farm and farm-specific risk factors, of course, regarding to wild birds, then the knowledge, awareness, and training of the staff working at the farm, the structural conditions, staff and vehicle movement, materials, animal movement, animal health monitoring, and so on. So into these 7 categories, the risk factors are attributed to. So just in a nutshell, what we're talking about. And now let's have a look at the tool itself, ladies and gentlemen. So it's free of charge. So you go into the internet on this website. I can also share the link afterwards in the chat. And then you select here the language you would like to work into, and here you would then go to the AI risk traffic light. So then you get some instruction how you can use it, and then you press start risk analysis, and then you go to the next page. Here I just selected here Sweden, and you will be asked if it's your first time here with our farm risk tool, or second time, or if you just have interest to look at the tool. Then you select what kind of species, so turkeys or broilers or geese or whatever, and then you enter the number of animals and stables or houses, and this is the only information we will gather. There's no IP address or no any other information from you taken into account. This is just for general information for us, but the information is not saved to our system. You will have in the end, I will come to this later, the opportunity to save your data as a PDF for your own documentation, but currently you do not have the possibility to log in again, because then we would need to attribute a password to you, and then there could be some linkages, and we just want to be really clear and transparent that there's no saving of any, that we do not save the data actually. Okay, and then it's a very interesting thing because especially here in northwestern part of Germany, we experience some times that we do not have internet. Instead of 5G, we have 0G. So then you can do it also offline. So you can save the questionnaire as an XLS document. And once you completed the questionnaire, you can upload it, and then you can also get the risk assessment. So I will just show you how it looks like. So it's not that a nice one, but you have a plain XLS file where you where you'll get some information. When you scroll down, you see all the risk factors, and then you can select the answers according to the questions. So all the so just for your information, so that this is also possible. But when you go on the online system, you will have about, as I mentioned, 100 questions, and there is a multiple choice questions. So when you can select the answer you would like to give, and we also vary the position of the answers, because we experience that some farms of an integration, a well-known European integration, they just click through for every farm, and then they tend to have very good results. However, we now kind of make it a bit dynamic, so that you really need to think about, okay, what is the correct answer for my farm I'm referring it to? Okay, and you just click through all the different questions, and when you then come to the end, you directly get the results. So then your farm is assigned to a risk category from 1 to 3 based on the total score from the online questionnaire. So the more risk factors identified on a poultry farm, the higher the potential is the risk of pathogen or AI introduction. So, and here you can then see the traffic light. It indicates, in this case, it was attributed to risk class 3. And then you have the possibility here to get the overall assessment, or you just have a look at the top 10 risk factors, or you just wanted to view the risk analysis only, and you can save all the information as a PDF. So when you click, I would like to have the PDFs, you can see again your results. And on the next pages, you will then have a look at all the risk factors that apply only to your farm. So based on the epidemiological relevance, so the risk factors with the highest priority are ranked first, and then all the other risk factors are listed here with your answer, with some optimization suggestions and some to-dos. And you also have here the possibility also to take notes and document it as you like, digitally, or you just print it or save it. Yeah, this is actually the tool. So just a quick information for you. I would like to draw your attention to the fact that this is really a non-commercial project without funding. So we got funding for programming it, and we got funding from the industry and also from our local government. However, this is a long time ago, and I do not have any staff who's taking care of it. So it's just a very intrinsic motivation to keep this program going. So if you imagine to find any bugs, or if you have any hints for further improvement, please kindly send it to me, because it's very, very valuable to get your feedback. And I just realized that the English version, that there are some terminology that a student of myself wasn't aware of, so I will also adapt it when I have time. So it's an ongoing process. So you're very much heartily invited to come to me if you find anything that could be improved. Okay, yeah, coming to my conclusions and the outlook. Well, we also ask a lot of times, what kind of users do we have? So we have a lot of farmers, of course. We have agricultural businesses, so vertically integrations, for example, our very good partner Wiesenhof, for example, or we have we work with OSI at a supplier from McDonald's who also uses this tool, or a lot of slaughterhouses from the pig sector, for example, working with the ASF system. Use this tool, but a lot of veterinarians and consultants, so some veterinary practices are kind of power users, we call them, because they really use this tool to structurally consult the farm. So they go to the farms, they clearly use it to have a structure for their farm visits, but also, of course, other stakeholders along the value chain use the tool. Yeah, and you can use it for your own internal farm assessment, but for your regulatory documentation, of course, but also for training and consulting support. And I just brought some very few data. We, of course, publish a lot, and we, with our partners from the Friedrich Loeffler Institute, with the Institute for Epidemiology in Reims, we are closely collaborating, and we did a small study where we just selected the farms in Lower Saxony that used the questionnaire from August 22 to December 2023. So, and we have, as you remember, these 3 risk classes. And when we look at the results, so we had in this time frame, we had 470 uses, and like more than 60% were attributed to the lowest risk class 1, 1/3 roughly was attributed to risk class 2, and to risk class 3, almost 10% of the farms were attributed to this risk class. So, and this, ladies and gentlemen, is of course the highest risk for further spread, as Jean-Luc was also referring to. And when you look at the map of Germany, and this is a map showing the poultry density by on NUTS3 level, you see that here in the dark brown area, here is Lower Saxony, where we have the highest poultry density next to the Netherlands and Europe. So that means if we have here farms that are attributed to risk class 3, this is really a risk for further spread when we have neighbors that really don't care about biosecurity, for example, especially in terms of an outbreak of avian influenza. And this would be the case also for Brittany in France, or the Netherlands or Belgium, or the Emilia-Romagna region in Italy, or in Catalonia, where we have some really regions where we have high density of poultry. But well, so when looking at the data, what are the top 5 risk areas, what we currently do see from the answers when we go to the farms? So the first one is that the consequences of daily operational routines, then that the hygiene logs, so the separation between black and white, is not sufficiently continuously implemented. But also the farm staff, we often have, especially in times of Corona, so COVID-19. So in times of COVID, we had a lot of staff shortages, but also language barriers, where the protocol, what needs to be complied with, is not sufficiently communicated to others. To the staff probably temporarily working at the farm. But also if you do the catching, if you do a pre-catch, then there are a lot of risks attributed to these procedures, but also the carcass disposal. So, and I just brought only some pictures. So what we every time see, and I'm really traveling a lot. This is Eastern Europe, so Hungary, for example, but this is also Germany. Where the carcass container is not cooled, is not cleaned, is next to the poultry house, is not working, I don't know. But there are also a lot of very, very good examples. So they were directly implemented in the fence. We also have the case that the farmers put a little window inside, so that you do not need to take the dead birds and carry them out with your clothes that you're wearing in the poultry house, but you can directly throw it through this little window here, and then after you go through the poultry house, you change your clothes, and then you take the dead birds and bring it to the carcass container. But well, just some examples. I'm happy to show more pictures afterwards, but referring to the time, we need to go on. So, ladies and gentlemen, we have a new area of responsibility for the farmers. So we have, of course, the economic part. So you need to protect your livestock and your operational continuity. You have a legal obligation, especially the compliance with the animal health law, but you also have a structural responsibility to contribute to animal health and the integrity of your whole value chain. And probably the most important one, your ethical responsibility, because if you do if you comply and are committed to biosecurity, this is applied animal welfare. So please take this or consider this, these different areas of responsibility. So I will skip this regarding the animal health law, but I just wanted to draw your attention to the economic consequences that this is just for Lower Saxony in the years 2020 to 23, that we have a lot of costs attributed only in Lower Saxony with outbreaks of avian influenza. And when there is an outbreak in Germany, and probably also in the other EU member states, you get, of course, some funding. So from the Animal Disease Fund, from state funding and European co-financing. However, our Animal Disease Fund is now implementing, also regarding or in close correlation with the animal health law, a scheme so that your compensation will be diminished when you do not comply 100% to biosecurity. So will ask you, in case of an outbreak, how likely is the infection to be introduced or spread due to violation to your biosecurity concept. And you see here that it can really, really harm your compensation when you do not comply to the biosecurity status. And just to give you some example, or one example, so only the medium, so when there is a medium likelihood that you have not complied, for example, when you store your straw or your bedding or what else, and that should be, that is used inside the barn, a lot of times it is placed outdoors. And when you place it outdoor or you store it outdoor without sufficient protection from wild birds, you might be only compensated 50%, just for you to know. So just as an example, what we do here in what the farmers and that we are facing here in Lower Saxony in Germany. So coming back to the tool, ladies and gentlemen, so what can it do? So actually it is a systematic identification of risk factors. It identifies operational weak points, and it gives you some suggestions for improvement, and can support training and advisory purposes, and it may complete also existing biosecurity concepts. However, there are also some limits. It's not suitable for purely pasture-based systems, or sort of like hobby farming or mobile egg producers. We're currently working on a new farm risk tool, but this is currently only for professional housing systems. It is also not an early detection tool, but a preventive instrument, and it requires really time and your honest answers and your intrinsic motivation actually to optimize your biosecurity. So, and what we also do face is that regarding to climate change and the emergency of vector-borne diseases, as well as our societal shift towards open barn systems, we really, it really requires a full and continuous personal responsibility. So prevention remains a cornerstone of animal health, welfare, and also the production security. So our traffic light or risk traffic light or farm risk tool, as we call it nowadays, is really helps you for a structured assessment. It enables a farm-specific risk evaluation, and it can serve also for animal health law-required biosecurity plans. But of course, the successful implementation requires joint efforts through all throughout all the value chain as a cooperative action. So this is my actually my last slide. So discipline actually is everything, right? So it doesn't count if you just clearly perfectly clean your boots, as you can see here, and then your then you put these boots outside where a lot of migratory birds fly over, and we a colleague of mine visited this farm, and we saw the cleaning and disinfection of these boots, and afterwards, and when we come back from our audit, it was complete shedded all over from migratory birds poo. So this was very nice example. However, so prevention is a duty. So you as a farm manager are an entrepreneur, but even the best and most comprehensive biosecurity plan really develops serious weak points if, for example, if you just quickly during the evening and before the evening news, you rush without boots and appropriate clothing in the barn just to check, or if the farmer tiptoes through the anteroom, but also wearing his boots and clothes from the premises. So also, we need to take into account when we go through wild bird resting areas, lakes and peatlands areas, hunting activity is just quickly is a problem, and staff shortages, language barriers. So biosecurity is really an individual, societal, and shared responsibility of everyone involved in the value chain. Yeah, and I just would like again to thank Natàlia and Ingrid and Julia a lot for this award we were recognized with. So we have been the BroilerNet champion. So it's very nice. Thank you very much. And we were honored to meet you all in Valencia last year in beautiful Catalonia. So thank you very much for this. And yeah, I'm glad to take any questions if there are any. So thank you for your attention.
Thank you very much, Barbara. Excellent. Before I ask Lina for the questions that are specific for your presentation, I would like to take the opportunity to ask a question. Long ago, I was a teacher for vet students where we were doing farm investigations. And at one farm, we the students came up with 25 or 30 action points. And then I had a follow-up with the farmer. And then he said that, oh, there were lots of good action points, but there were so many I didn't really have the courage and the energy to perform anything. Is there any guidance of how to make a priority? Because Rome might not be built in one day.
And you're so right. This is also a psychological effect, right? So we also had this several times, and that is why we also did this. Just look at the top 10 or top 10 risk factors at the farm, and of course, sometimes it's really overwhelming when you get this assessment and you see, wow, where to start, and yeah, actually, it's every time good to talk with somebody with an external view who helps you probably to prioritize, for example, with your veterinarian or with your consultant where you have a trustful relationship with. And then, as you mentioned, Rome is not built in one day. So it's really just take probably first 1 or 2 risk points, and then you working yours throughout. You can also do a draft, an action plan, but sometimes it's really good to have some external expertise or external support friendly support who really helps you to guide through it. But yeah, but actually now the pressure is there that we need to take action, especially these animal health law things really struggles because we have there are a lot of things that going on. So from next year on, if this biosecurity plan and the risk assessment is not in place in Germany, they will also lose compensation. So that is really that is why the farmers are currently really forced to do something. But they also can get a compensation for consultancy. So I don't know how is it in other countries in Europe, but this is very nice, so that you really can get help to get you through this procedure.
Thank you. Lina, are there any questions that are specifically for Barbara?
Yes, there's one question that has like two parts. So the first is, how many farmers in how many countries have used the tool? And the other question is, could you use the tool for research and, for example, do follow-up on red farms to see if they have done any of the interventions to improve?
Yeah, thank you so much, Nina. So starting with the last question, yes, we do research together with the Friedrich Löffler Institute, the Institute for Epidemiology, that we really focus on these red farms and try to evaluate it. However, as the data are anonymously, it's really difficult to dive into it. But we can of course do some nice statistics and nice evaluation. And regarding the first question, currently we have more than 5,000 users for the avian influenza tool since 2018. But there are a lot of power users, so I can not directly tell, because a lot of power users download the XLS file and then upload, or probably do it manually. So there might be some data we cannot access. Regarding the other species, I would need to look up, but it's really good. But of course, the main focus is currently still in Germany and Austria and the Netherlands. But we really hope, together with this seminar, a webinar, and any other conferences where we present the tool, that it will be also used in other countries.
So now we have like 8 minutes, because I will round off this 1 or 2 minutes before 12 o'clock. But now it will be questions for the whole panel. So Lina, what can you offer in questions here?
I can offer a couple of questions about the situation in the US. So one is, what do you think about the US and their current bad situation regarding the position towards a positive position starting vaccination?
Who would like to answer that question? Jean-Luc looks like he wants.
Or Jean-Luc. Jean-Luc, what do you think? Yeah, you can answer.
Yeah, let's say the situation is still concerning, obviously. Still, we observe a lack of control, to be honest, a lack of control in the poultry sector, and even more in the cattle industry. Yeah, that's the situation. We learned some ideas of stop any strategy of control, which is completely nonsense. Okay, but hopefully it will never be implemented in the field. And regarding the vaccination strategy, I think that the impact in the layer industry is so important that the question it must be on the table, and globally, not only in the US, to be honest, because we observe some cases, some outbreaks in huge farms, and the economic impact is huge. So the question of the sustainability, depending on the evolution, specifically on the layer industry, is on the table. I would not say the same regarding the broiler industry, given the very short production cycle. But regarding the layer, I think we can discuss this. That's my position, Natàlia.
No, I fully agree with you. And actually, I mean, since the beginning of the year, the information that we have is really scarce. I don't think that they are, I mean, giving all the information on what's happening in the farms, in the cow, in the dairy farms, and in the poultry farms. And again, yeah, I agree that this some opinions given by the authorities, which is really alarming, are not nonsense at all. And if they consider vaccination, and I think that also that in layers it would be maybe indicated. It has to be coupled with a monitoring system, with a very hard monitoring system and surveillance system. So, yeah, we'll see. But the situation is complex, and I mean the news that come from there are not positive at all.
Thank you. Do we have another question, Lina?
Yes, there's one. What about the current H5 surveillance in dairy cattle in Europe?
Natàlia,
Yeah.
maybe, or?
Well, as far as I know, there are no surveillance program. There is no other surveillance program in cattle in Europe so far. I think that, well, maybe in some parts people is, I mean, looking for if there are positive animals in some farms, but it's not, I mean, like a governmental surveillance plan that is established.
Huh? Another question, Lina?
Yes, there's another question about vaccinations, which is about small farms versus big farms. I'll read it just as it says. What about vaccinating a small family production in so many villages? Big farm production will do it, but the small ones will not. I'm asking this because the virus transmission.
Yeah. It's also always a difficult issue. We know that the compliance with good practices of vaccination and even more surveillance, it's much more difficult in very small farms. That's why, for example, in France, the threshold is 250 birds. Below this threshold, there's no vaccination, because we consider that they are not really commercial farms, and it would be very difficult and very costly to implement vaccination and surveillance in so small scale farms. That's also a balance. So we have to consider a balance, and we have to consider that vaccination is not the only tool. We have to associate vaccination, surveillance. So we could adapt some specific tools for this type of farms, but I'm not convinced that in this case vaccination is a good tool for this type of farms, to be honest, because it's not cost effective to proceed like this. It's too risky. We risk to have a very poor surveillance, and I do not think that that should be the priority. I understand, because we know this, even in France, we have some pressure from small producers that are actually excluded from the programs. But we have to keep in mind that vaccination is not the only tool. It's you have to consider the global set of tools, and we should focus surveillance, specific surveillance, and biosecurity on these small farms. At least it's my own opinion.
Do we have a final question, Lina?
Yes, a final short one. What would be the average or maximal distance for a farm-to-farm airborne transmission of the virus?
From practical point of view, Barbara, do you have an estimation here?
Sorry. There are different papers and different publications currently. Uh, I would like I can't give a clear answer here. There are different opinions actually on that because there are different papers and they all contradict each other from my point of view. But probably Natàlia and Jean-Luc want to give an answer here. But it really depends also on the where the farm is standing, what the wind, where the wind goes, and it's really different aspects to consider to give a clear answer here. Natàlia probably would like to add to that.
No, I don't have the clue. I guess that there are, there are different papers saying different things, and I think it's not clear. I guess it's not the main route of spreading the virus among the different farms, but it may play a role. But I don't think there is like a distance that you can say, I mean, this is a safe distance or not. I don't think this is established.
Thank you very much. It's 12 o'clock now, and I would like to thank you all three speakers for giving us excellent knowledge from different positions. Barbara, Natàlia, Jean-Luc, it was nice to have you on this webinar. And I think there are still some questions, but officially I will close the webinar, and I thank you and congratulations to be the BroilerNet champion. And we will have future webinars with other topics related to the champions within BroilerNet. And I also put in the chat the link to our LinkedIn account, and you can follow us there and see what would be interesting for you, your production or your work as veterinarian or advisor within the broiler sector. So thank you very much, and I hereby close this webinar.